بسم الله الرحمن الرحيم. السلام عليكم. The title of our lecture is transplantation, the immunology of the transplantation. The objective of this lecture is to describe terms used in transplantation and tests used before seeing patients for the operation of the transplantation, and the immunologic mechanism and types of rejection, treatment, and also the tumor immunology. Here we have some terms that's used in transplantation. Autologous, which is mean transfer one organ or one tissue or one cell from one part of the body to another part of the body. And usually this is occur in the skin graft. And the other term, which is syngeneic, which is transplantation of the graft or tissue from one person to another person, which they are genetically identical, which is occur in the identical twins. Here we use a graft, the term graft, for any organ or tissue or cell which is transplanted from one person to another or in the same person. And this is the term graft. And the syngeneic is the transplantation of the graft from genetically identical twins. And allogenic, which is very important, which is a transplantation of the graft between two different individuals, which they are genetically different, but but they are in the same species, يعني إنسان الإنسان. Xenograft, which is transplantation of the graft between two different species, from the animal to a human. الآونة الأخيرة سووا transplantation of kidney from the pork من الخنزير، ولكن فشلت شهر وتوفى المريض. Causes of rejection. Recipient ليش يصير عندي rejection؟ A recipient immune system looks to the graft as a foreign substance. So this leads to stimulate the immune system. And how this stimulation occur through the histocompatibility antigens, which is present on the cell surface of the cells. The immunology of allogenic transplantation. First, cell-mediated immunity. So the part of the immune system which is responsible for the rejection is the cell immunity, which is important cell is T cell CD4 plus, which is stimulated due to exposure to the allo-antigen, which is from the donor plus the MHC. This occurs through two phases: sensitization phase, with the antigen reactive to lymphocytes of the recipient, and these lymphocytes will proliferate in response to this allo-antigen plus the MHC. And the after sensitization phase, there is an effector phase, which is aimed in the extraction of the graft or the tissue which is transplanted in the patient. So the patient must be sent to the following test before do the operation. First, the ABO blood group, tissue typing, which is HLA-B, C, G, and HLA-DR, BQ. Second, another test, which is a cross-matching between donor lymphocytes and the recipient serum. Another test, which is a frozen lymphocyte array, FLT produced for T and B cells for detection the antibodies in the patient's serum, and this is monthly done. And it's called anti-HLA antibody screening. And last one, C4D staining, which is deposition of C4D. This is the complement C4D in the graft capillaries. Another test, which is virological test, cytomegalovirus. As we know that the target organ of cytomegalovirus is kidney, and the Epstein-Barr virus, the hepatitis B, C virus, and the HIV AIDS virus. Types of donor. In Iraq, the relatives, either father, mother, sister, brother, while abroad from the cadaver, from the car accidents or any accidents, and patients with brain death. And there is a European Association, which is European Association for the Transplantation, which is include all countries in Europe. And the another association, which is Scandinavian Association, and this includes all Scandinavian countries, and the American Association for the Transplantation, and this is include the two Americans, North and South America. And abroad, the patients with, for example, renal failure, or any organ failure, because only in Iraq we have renal transplant, while abroad they have the liver, lung, heart, bone marrow, etc. So the patients still on the waiting list, and there are scored for the priority of the patients, which is range from 0 to 10 points. If the patient had HLA matching positive with between the patients and the donor, so had 2 points. And if the patient HLA reactive antibodies negative, had 2 points. And the waiting time more than 5 years, so the patients had 2 points. And age of the patients, children preferable than adults because of the erythropoietin, which is secreted from the kidney. And patients who had cytomegalovirus negative, so had 2 points. So patients who had more score, more advantage for doing transplantation. And for example, as we said, renal patients with renal failure, and the operation done for him, and this is the transplanted kidney put in the right iliac fossa. This is anastomosis between artery and vein, and the ureter with the bladder. So if it is accepted, it's thank for God, and it's okay. And if there is a rejection, so we have types of rejection. Hyperacute rejection, which is occur due to preformed antibodies, and this antibodies formed due to previous blood transfusion or previous transplantation, or due to multiple pregnancy. Here the mechanism, this is the antigen in the blood vessel of the donor kidney, and the body will form antibodies against these antigens, and this leads to complement fixation due to antigen-antibody complement fixation, and lead to attraction of neutrophil and formation of microthrombi, which is lead to necrosis. And acute rejection, which is a process of vascular and parenchymal injury, which is mediated by T cells and antibodies, which is begin after first week of transplantation. And in a hyperacute rejection, so this is occur within minutes. The patient is still in theater, and rejection occur in operating theater. While here, after weeks or months, the